Carbohydrates. Carbohydrates are made up of carbon, hydrogen, and oxygen. There are simple carbohydrates, which include monosaccharides and disaccharides, and complex carbohydrates, which are polysaccharides. On the left, we have glucose and fructose. These monosaccharides each have 6 carbon atoms, 12 hydrogen atoms, and 6 oxygen atoms, but their structure differs. Note the aldoses and ketoses in the left and right images on the screen. Aldose carbon skeleton is in the aldehyde group, and ketose in the ketone group. Then we have the middle image, which is ribose. Ribose is a pentose, and to the right is glyceraldehyde and dehydroxyacetone, which are trisoses. Here is alpha-glucose and beta-glucose. The hydroxyl group joined to the first carbon is switched around. Alpha-glucose monosaccharides will form covalent bonds and orient the same way. However, beta-glucose have to alternate upside down to its neighbour to bond. Lipids. These lipids are composed of carbon, hydrogen, and oxygen. 2 forms of lipids are triglycerides and phospholipids. Triglycerides are made up of 1 glycerol and 3 fatty acid chains. Phospholipids have a similar structure but only contain 2 fatty acid chains. Phospholipids have a hydrophilic polar head and hydrophobic nonpolar tails. The fatty acid chains are connected to the glycerol by ester linkage, a bond which is formed when a hydroxyl group joins a carboxyl group, which you can see here in red. Fatty acids can be saturated and unsaturated. Saturated carbons have formed their maximum number of bonds, whereas unsaturated have double bonds and less hydrogen atoms. When a double bond occurs, it can cause a kink in the chain. Lipids are not true monomers. However, they do go through a similar process of smaller molecules joining together and performing a condensation reaction. Protein. Proteins consist of an amino acid group, a carboxyl group, and an R group, also known as a side chain. A protein consists of 1 or more polypeptides which are formed and coiled in a 3D structure. Polypeptides are polymers of amino acids joined together. Polypeptide bonds are formed through condensation reaction. 4 levels of protein folding are primary structure. This is the amino acids forming a unique linear sequence structured by genetic information and held together by peptide bonds. The secondary structure sees the amino acids folding according to their sequence due to a partial positive change from hydrogen, which are attached to nitrogen, and a partial negative change from oxygen. This charge enables hydrogen bonds to form between them, resulting in folding and coiling. Here on the screen is 2 ways this can occur. The coil shape is the alpha helix, held together by hydrogen bonds. The formation on the left is the beta pleated sheet, formed by 2 or more polypeptide chains lying side by side. The tertiary structure is where the R group interactions determine the folding shape. R group interactions include ionic bonds, disulfide bridges, hydrogen bonds, hydrophobic and hydrophilic reactions, and van der Waals interactions, which helps hold the shape together. A quaternary structure is a protein's final form. This example on the screen is a collagen protein, a fibrous, large triple helix. This gives collagen its strength, and 40% of proteins.
A biomolecule is an organic molecule that is produced by a living organism, including large polymeric molecules such as proteins, polysaccharides and nucleic acids, as well as small molecules such as primary metabolites, secondary metabolites and natural products. Lipids are an important part of the cell membrane. They also play a role in serving as a source of stored energy and acting as chemical messengers. They are made up of glycerol, 2 fatty acids and a phosphate group. They contain carbon, hydrogen and oxygen and are made of hydrocarbon chains and contain fewer polar hydroxyl groups. This means they do not dissolve well in water. Proteins are major structural elements of cells. Their role is to transport nutrients in and out of cells and as enzymes and catalysts for most chemical reactions that happen in living organisms. There are 20 different amino acids that occur in proteins. The order of which the amino acids occur plays an important role in determining protein structure and function. Carbohydrates are made up of molecules containing atoms of carbon, hydrogen and oxygen. They are essential energy sources and structural components. They are made up of 4 types of units, which are monosaccharides, disaccharides, oligosaccharides and polysaccharides.
Formation and breakdown of polymers. Polymers are broken down into monomers through a process called hydrolysis. A polymer is a substance or a material made up of very large molecules or micromolecules composed of many repeating subunits. Some natural polymers are formed with one kind of monomer. However, most natural and synthetic polymers consist of two or more different types of monomers. These polymers are also known as copolymers. Polymers are formed through a process called polymerization. This is where monomers chemically react together to form a polymer chain. Next, we move on to the external factors in the enzyme activity. As shown in the image to the top left of the screen, there are many effects of external factors in enzyme activity. Here I have outlined 3 of these. The first one is temperature. All enzymes need an optimum temperature in order to work properly. Enzyme activity increases as the temperature rises, and in turn this increases the rate of reaction. Next, we have chemicals or drugs. Chemicals and drugs are inhibitors. Inhibitors alter, slow down, and in some cases stop catalysis. Inhibitors also block or distort the active site. Finally, we have pH values. Each enzyme has a range of pH values called optimum pH. When the pH value of the reaction changes, the shape and structure of the enzyme will also change. When this happens and the temperature rises too high, the change in the active site is permanent.
So here are just some facts about what enzymes are and what they do. So enzymes create the conditions needed for biochemical reactions to happen faster. So they do this by lowering activation energy. So enzymes can help break something down, which is called a catabolic reaction, but they can also help build something up, which is called an anabolic reaction. So enzymes are also known as biological catalysts because they catalyse the chemical reactions that happen inside living things. So here are just 2 examples that I thought was easy to simplify the structure of enzymes. So if we're referring to the right hand side, what will happen is the enzyme will meet the substrate will then climb aboard the enzyme. So then the enzyme substrates will wiggle around, which will be forming a new bond between the substrates. Once that new bond has been formed, the enzyme will release the product of the reaction, which as you can see has been shown in both diagrams. So how the structure of enzymes relates to its function. So the shape and structure of an enzyme is really important for its function. The main structure of the enzyme will determine which reactant will bind to it to form a reaction. So any deformation in the structure of an enzyme will prevent the reactant molecule to bind to the enzyme and form a specific bond. So here on the right hand side is a perfect example, and this is a famous model called lock and key. So in this example, the enzyme is the lock and the key is the reactant molecule specific to the enzyme. So the reactant molecule will fit in the enzyme properly. So an optimum temperature and pH are important for the enzyme to function properly. So any changes that may affect the structure of the enzyme, such as the temperature, this may affect the structure of the enzyme and the activity, which gives the enzyme the ability to function. So basically what that means, so the reason why the structure of the enzyme relates to its function is because if there's any sort of deformation within the structure of the enzyme, it will prevent the enzyme from being able to function properly, which is why the temperature, the pH, the shape, the size, everything always has to be specific to the enzyme in order for it to function properly.
Concept of activation energy. So activation energy is the amount of energy the enzyme needs to produce to allow a reaction in the substrate to occur. This reaction coordinate graph shows how much energy input would need to be put in for a reaction to occur. The graph works out the free energy of activation by measuring from A to its transition state at the top. This measurement is the amount of energy A needs to use to get to B. See the difference between a non-catalyzed and an enzyme-catalyzed reaction.
The lock and key theory was proposed by Mayne Fischer in 1894. In this analogy, the key is the substrate and the lock is the enzyme, meaning that only the correct substrate will fit in the enzyme. The enzyme-substrate interaction suggests the enzyme and the substrate possesses particular complementary geometric shapes that fit precisely into one another. Any other substrates will not fit in the enzyme, only the correctly shaped substrate will. The lock and key model does not need a separate catalytic group for the action of the enzyme, and also the active site of the enzyme contains the sole entity. the active site of the enzyme is static and there is no weakening of the substrate bonds involved. The induced fit theory was suggested by Daniel Koshland in 1958, where the theory assumes that the enzyme is flexible and the substrate helps determine its final shape. When the enzyme has been distorted too much, certain compounds combine but will not react. To determine the final shape, the enzyme and the active site will continue to change until the substrate is completely bound to the active site of the enzyme. In the induced fit theory, the active site of the enzyme contains two components, and unlike the lock and key theory, there is a separate catalytic group in the enzyme. The catalytic group weakens the substrate bonds either by nucleophilic or electrophilic attack, and the active site is not static.